Have you ever stopped to think about what really makes you, well, you? It turns out deep inside your head is the most complex object we know of in the entire universe. It's this incredible web of connections that holds all your thoughts, your memories, your entire being. So today, we're going to dive into this monumental scientific quest to create the very first map of this inner world, to finally read the brain's blueprint. At the very heart of this journey is a question that gets right to the core of who we are. Is the wiring in your brain something that was handed down from your ancestors through your genes? Or is it something that's drawn and then redrawn by every single thing you see and feel and learn in your life? It's really the ultimate nature versus nurture debate, playing out right inside our own minds. So scientists basically see this as two powerful forces working on totally different schedules. On one hand, you've got evolution. We're talking millions of years where our genes have slowly encoded a basic wiring plan. Think of it like a factory-installed operating system that's been tweaked for survival. But then, on the other hand, you have your own life. Every experience physically imprints itself onto that circuitry, customizing it in real time. The big mystery has always been, how do these two forces actually work together?
Well, to crack that mystery, a whole new field of science has sprung up, and it's called connectomics. And it is incredibly ambitious. The whole goal is to map every single wire, that's every neuron, and every single connection, that's every synapse, in the brain. It's kind of like creating a Google Maps for the mind, but with resolution so unbelievably fine you can see every single tiny intersection. Okay, but before we can even begin to read this map, we really have to wrap our heads around the sheer, mind-boggling scale of what we're trying to chart. This is not like mapping a city or even a country. This is like mapping an entire universe that's packed inside our skulls. To really get a sense of the scale, just think about this. The entire brain of a fruit fly, everything it needs to fly, find food, do its thing, that entire brain can fit inside a single column of a mouse's cortex. And that single column is just a tiny fraction of the whole mouse brain. The jump in complexity is it's on a whole other level. But here's the good news. Our technology is finally starting to catch up. I mean, just in the last 20 years, thanks to huge leaps in microscopy and artificial intelligence, we can now map a volume of brain tissue that is 1,000 times larger than what we could do before. It's this incredible technological sprint that's letting us actually keep pace with the brain's insane complexity. So what is it that makes this so ridiculously hard? Well, it all comes down to the axons. These are the long, skinny wires that connect all the neurons. They're about 100 times thinner than a human hair, and they're packed together so densely it literally looks like an impossible knot of spaghetti. The real challenge is trying to trace each individual strand from its start to its finish, without ever losing it or mixing it up with one of its billions of neighbors. And this right here just shows you the problem perfectly. What you're seeing is the connections from a single neuron in a mouse's brain. And look at how they just sprawl out across almost the entire thing. It means that if you want to map the complete wiring of just one of these cells, you can't just look at a tiny piece of the brain. You basically have to be ready to map the whole thing. So how on earth do you actually do it? How do you map this inner universe? Let's take a look at the incredible technology, the Explorer's Toolkit, that's making this whole journey possible. It really boils down to 4 key steps. First, you've got to prepare the brain tissue. Then you slice it into unbelievably thin sections. After that, you take a picture of every single one of those slices with a super powerful electron microscope. And finally, you use some seriously powerful AI to stitch all those millions of images back together into a full 3D model. That first step, preparation, it's almost like a form of alchemy. See, the delicate membranes of neurons are pretty much invisible to an electron microscope. So to actually see them, scientists have to stain the brain tissue with this cocktail of heavy metals. We're talking things like osmium, lead, and uranium. These metals basically grab onto the neuron membranes, and all of a sudden, these invisible circuits just pop out in sharp relief. But even just preparing the tissue is really tricky. In the past, the methods they used would kind of squeeze all the structures together, giving you this distorted picture. But modern techniques can preserve the brain's true geometry, keeping that natural space between all the cells. And getting this right is absolutely critical, because you want a map of the brain as it actually is, not some warped funhouse mirror version of it. So all of this incredible, painstaking effort, it is starting to pay off big time. Scientists are now getting their very first glimpses of these new maps, And they are bringing back some incredible treasures from this new scientific frontier.
A truly monumental achievement was announced not too long ago: the completion of the first full connectome of an adult fruit fly brain. It was this massive undertaking that proved once and for all that mapping an entire complex brain from start to finish isn't science fiction anymore. It's actually possible.
And this is what the data looks like. This beautiful, chaotic, vibrant tangle is a 3D reconstruction of less than a single cubic millimeter of a mouse brain. Every single color you see is a different neuron. This isn't just a diagram, it's the real thing. It's like a dense, tangled forest of thousands of cells, and now, for the first time, we can navigate through it, branch by branch. So what are these first maps actually telling us? Well, for one thing, we can now see exactly how evolution hardwired certain abilities, like the specific circuits a fly uses for detecting motion. We're also getting the very first precise blueprints for local circuits in the mammalian cortex. And maybe most importantly, these maps are starting to show us just how much of the brain's circuitry seems to be left open, almost like a blank slate, just waiting for us to fill it in with our own unique experiences. So this all leads to the big question, right? Where does this incredible quest take us next? What's on the horizon for these new cartographers of the brain? Well, the roadmap is incredibly ambitious, but it's also crystal clear. We started small, with tiny circuits and insect brains. Right now, we are smack in the middle of the era of mapping cubic millimeter-sized chunks of the mouse brain. The next great challenge is to scale that up to an entire mouse brain. And of course, the ultimate goal, the thing everyone is working towards, is to apply all these techniques to map huge sections of the human brain itself. And let's be clear, this isn't just some academic exercise. A complete map of the brain could totally revolutionize medicine. It could let us understand disorders like Alzheimer's or schizophrenia as what they really are, problems in the brain's wiring. It could unlock the secrets of aging. And hey, by understanding the brain's own blueprints, we might even learn how to build much more powerful and efficient artificial intelligence. Which brings us to the final, absolutely tantalizing question. When we finally have it, a complete synapse-level map of a human brain, what are we going to find? What secrets about consciousness itself, about how memory really works, and about the very definition of ourselves will this ultimate map finally reveal to us?